which means you take two sugars and you bond them together, hence disaccharides, right? There's a couple common ones. I won't ever directly test on that, which you should have seen already, that I didn't ask that kind of question. I did ask which of these is a disaccharide on the test, but then what did I list as three possible answers? All monosaccharides that you were required to know. So process of elimination, that leaves you with the last one. Okay. In future classes, you might very well be expected to know what sugars make these individual common sugars up. Okay, so kind of be aware of that when you're looking at your disaccharides. There's a list of a whole bunch of important ones. You get your sucrose, fructose, all that fun stuff. The structure and function of polysaccharides. So if we now take this a step further, instead of just linking one or two sugars with each other, we can actually build these longer chains. These longer chains of sugars can carry a variety of roles. They can contribute to structure and they can contribute to energy storage as well, depending on how it's linked and what sugars are actually used within it. So cellulose is one of those structural ones. So when you're looking at cellulose, it's linear, so we're literally just linking those sugars next to each other. And we're looking at a pretty massive weight. It says, or sorry, not a weight in that case. We're looking at roughly 3,000 glucose units linked together. They're also linked to that beta-1,4 linkage. Okay, so in theory, you will remember that referencing on how we're linking those together. So beta-1,4 means what? Beta means? We're looking at the anomeric carbon, and it's in the beta position, which means the OH is located where relative to the CH2? Same side, we're looking at those being cis. What is 1,4 referring to? Carbon number 1 is bonded to carbon number 4, right, through those alcohols. And that's all it's really referencing. That then continues to propagate outwards, and that's how we would build our cellulose structure. To build it and generate the kind of structural effects, you'll end up noticing a 180 flip on it. So depending on how they want to represent that structural drawing, you may see it with that weird curvy bond, or you might actually see the structure flipped over. Okay. One of the reasons why it's going to be used for structure is due to all the hydrogen bonding. So you notice with your sugars, you've got all those OH groups. All of those OH groups can then hydrogen bond to other chains. If we've got a nice long simple chain, that long simple chain can interact a lot with another long simple chain, and we can get a pretty strong interaction between them, and that can generate and help with our structural issues. Okay. So again, depending on how we want to represent it, so we take a look at drawing our Hayworth projections up top. You'll notice that they use that goofy curve bond. They're only using that so that we can maintain the Hayworth projection for each and every one of those sugars. Right. If we want to look at the actual structure, this is an awful representation of it, but if you look really carefully within this, you can actually see how everything is linked, and you can tell that they actually do some flips and rotations to align those bonds in the proper angling. Okay? So it just depends on that representation. The thing that's nice about their other drawing here is that you can see the hydrogen bonding effects, which they use with those dashed bonds. Okay, presumably they did them all correctly. It's those extra hydrogen bonding interactions that are really contributing to our stability of that. We can also shift into structures such as starch. Starch is going to be primarily your energy storage within plants instead of structural effects. We're looking at more sugar units going into this. This case we're looking at 4,000 as opposed to the roughly 3,000. We're still looking at that same linkage. It's not the same linkage. This time it's an alpha orientation around your anomeric carbon. That alpha change is then also going to change how that molecule can then stack and be organized, because you're now changing the relative position of those. All right? We get two primary different types. You get your amylose and the amylopectin. The pectin has cross-linking. That cross-linking is going to then make it a little bit more stable in the process of trying to take it apart. Okay, so it's going to be a little bit less accessible to degradation because you've cross-linked it, which is also going to contribute more structural effects. Instead of having now hydrogen bonding interactions, you've now physically bound those two chains, so you're going to generate a more rigid structure. One of the more interesting effects with your starch is that you can get these cyclic structures happening with your starch, and that cyclic system or secondary structure can wrap around iodine molecules. That changes the absorption spectrum of the iodine and the sugar molecules, and what ends up happening is you get a blue solution. Okay, so that's your standard characterization method for the presence of starches. Okay, you can add starch and iodine, and you'll see that nice blue color come out of that. Okay, from 152, you definitely use that in one of your experiments. When we go through to decompose these, we're going to use an amylase. One thing, I can't remember how many actual enzymes we've talked about so far, but you'll notice that ending there, the ACE ending, is typically used to imply a degradation. So you're starting to take apart something. So if we see an amylase or any kind of enzymase enzyme that's going to have an ACE ending, it's involved typically in some kind of cleavage. In the case of our amylases, what they're going to go through and do is actually take apart your starches. Okay? Because in that starch form, we can't actually process the energy. So we have to cleave off individual sugar molecules and then process the sugar molecule to get the actual energy out of it, okay? Your individual saccharide. There's a couple different types of amylases. You get your beta amylase and alpha amylase. What do the beta and alpha mean? Absolutely nothing. It means 1 and 2. Okay. So that's one of the reasons why I asked the question, is it doesn't necessarily refer to an anomeric carbon. Okay. It's just saying they're different types. So again, biochemistry doesn't have a consistent method in how we go through and apply things. It's just the way things work. Okay. So your beta amylase is what's known as an exoglycosidase. Exo meaning that it's going to start to degrade the polysaccharide by taking advantage of the sugars at the end of the structure. So you can really only remove one sugar molecule at a time, starting from the very end. Okay? It's a very slow formation of your sugar molecules, so we can end up converting all of those sugars across into your glucose. If you trust Wikipedia, it's only found in plants. Okay? The alpha amylase is an endoglycosidase, which means it can catalyze that destruction anywhere, that cleavage anywhere within the sugar or the polysaccharide structure. Okay? The thing that's nice about that is that we don't have to start at the ends. We can start anywhere, which means you're probably going to look at a much faster reaction, okay? Because we can start to cleave and break that into smaller chunks very, very rapidly. The more chunks we have, the more enzyme can get in there to potentially degrade it. Okay, so your alpha amylases tend to be a lot faster because they aren't as selective. They aren't forced to work at the end. Okay? And you'll get the same degradation products. One big issue with both of these amylases is that they cannot catalyze the glycosidic bonds found within the amylopectin. The pectin had cross-linking. So instead of doing our standard linkages, which, oh, actually this did there, never mind, I didn't have to go backwards. Our alpha 1,4 linkage, okay, when we cross-link in the amylopectin, we're doing beta 1,4. Did I do that right? Yeah, I didn't think so. Alpha 1,6. Your alpha 1,6 link cross-links, sorry, I keep skipping back and forth, of the amylopectin. Okay? When you're looking at the branching off to actually link multiple chains together, okay, the amylase can't cleave that bond. Okay? And that's because the geometry of the atoms around it is a lot different in the 1,6 linkage versus the 1,4. Okay, so what ends up happening is our amylases can't break that down, and we'd end up maintaining that structure. If we still want to get the sugar molecules out, we're going to have to use a different enzyme, and that's where we can use a debranching enzyme, okay, to catalyze that hydrolysis. So I believe the textbook actually lists a name for one, and it doesn't look like I gave the name for one just because I thought it was kind of lame to just list multiple names. We'll use some debranching enzyme. Okay, so all it's going to do is break that extra branching linkage. We don't get starch in animals. So how do animals store? Oh, it's a good thing I stepwise this. So how do animals store their energy? Okay. We're still looking at some kind of storage mechanism for our sugars. So we can get fats just like plants do. We get fats and oils are ultimately the same thing. We're looking at triglycerides. How can we store sugar molecules? Answer might be at the very top there. Glycogen. Glycogen is very similar to our amylopectin. We're storing those individual sugar units within a larger chain structure. Okay? It is highly branched, which is why it's comparable to our amylopectin. And we're looking at 24 to 30 units of the D-glucose in our branches. Okay? Every 24 to 30, sorry. You'll notice that we're going to have the same polymer backbone as our amylopectin, the 1,4, and we've got our branches coming off at the 1,6. So if it's giving us the exact same kind of bonding pattern, why don't we refer to it as amylopectin instead of glycogen? What's that? Weren't they all still alpha? They're all still alpha. That would have been a reasonable guess. I would have liked that, actually. Why? Animal versus plant is the best thing I can come up with. To me, this is, again, one of those freaking irritating things, okay, with biochem. We've got virtually an identical structure, and we're going to call it a different name just because it came from something else. Okay? There is slight variations. We're looking at 24 to 30, every 24 to 30 sugars to get your branching. Where did I list it here? Wait a second. It's not the same. We get more linkages. Let's see what else I have in my... Yeah, we get more linkages. I must have copied and pasted wrong. Okay. There's more branching in glycogen. Okay. And so that's its primary difference between that and amylopectin. That and its plant versus, or animal versus plant. Okay. Your other big difference is how it's broken down. It's not broken down by beta amylase, okay, but it's broken down by the equivalent enzyme as beta amylase. Does the exact same thing. It's going to be cleaving in the exact same spots, but of course it gets a slightly different name, okay, because it's being processed by something slightly different coming from a different animal source. Okay, so every protein that we get, every enzyme that we get, as long as there's something different about it, of course it has to have a new name, even though it does the same thing. Okay? We can look at other polysaccharides. We've got chitin. Chitin, again, we're going to change the structure around ever so slightly. So instead of dealing with just your standard sugar, we're going to introduce at the 2 position an amine. So we're looking at some kind of aminoglucose. That's going to be taken a step further and processed into the amide, or an amide bond, which of course we're then going to refer to as an acetyl as opposed to a legitimate amide, because we have an acetyl group attached to the amine. Okay, so yeah, it sort of makes sense. That change in the structure is going to change how the sugars can then hydrogen bond interact with other sugar chains. And that's what's going to contribute to this type of polysaccharide having a much larger difference as far as its properties. And we're looking at this adding to kind of exoskeletons, so structural features, not energy storage. Okay. You can look at cell walls, generating, again, more polysaccharides to go through and process. You're looking at, which ones were these? spend too much time on DNA. We're looking at your N-acetyl glucosamines, or your NAGs, and your N-acetyl muramic acid. I said that right? So again, we're slightly changing the structure of the sugar by changing one of the alcohol groups. And you'll notice that what we end up changing is the second position in our carbon chain. Come on, there it goes. So we're really changing that second position in both cases. The muramic, whatever that is, acid, our NAMs, also have a change out off of our third position. Okay. Those changes are now going to facilitate stronger interactions and stronger cross-linking between these. That's going to help generate those cell walls and allow us to actually build a more stable structure and prevent our bacteria from being crushed. So that's what our cell wall is going to help us protect for. We also have the Glycosaminoglycans, more sugars. Sugars involved in a lot of general processes. We can look at it as part of our immune system. So they've got all sorts of other contributions from those other sugars. So we could also look at all of those. Ultimately, it's just another list of all the possible ways that we could put all these sugars together. And those slight tweaks to the structure is going to change those properties a lot. Okay. Last one, we could look at our glycoproteins, which is where I was going with the amino system. Where can I cough? Our antibodies are all glycoproteins. So that's going to be a huge contributing factor to our immune system. One of the big case studies that we could look at would be different blood types. So your A, B, and O are all due to different sugar chains hanging off of the structure or hanging off of individual blood cells. Okay? If that antibody is different, if we have a different sugar sequence changing off, it's going to respond differently to our immune system. Okay? That response is why if you have A-type blood, you can really only have what type of blood transfusions? A. Or OYO. It doesn't have a receptor that matches that, so there's nothing to connect up. Okay? So it's that sugar connection, that antibody that's hanging off of your blood cells that's really being detected, and that's what's going to determine our ability to accept different blood transfusions. Okay? So it's kind of neat in that respect. So, and that's all, again, dependent on those saccharides. Questions? Pretty sure that's the last one. I already answered all the questions you could possibly ask about it, I guess, on sapling, so should be okay. Okay, so what we'll now do is push into DNA. So we've kind of done all we're really going to work with when we look at proteins. So we're going to look at our DNA and RNA processes. Before we can really get into that, we have to understand something about their structure. Just like we do with proteins, we know something about the amino acids and how those could be put together. So chapter 9 is all about the nucleotides and in general how they interact with each other to generate the differing structures. So your primary, your secondary, your tertiary structures. After that, we move into chapter 10, which is looking at DNA replication. Excuse me. And then 11 and 12, and I don't remember the order on which one's which. We'll probably do, what's it going to be? Probably transcription first and then translation. And we'll talk about those, again, looking at all those individual pieces. And then we have exam 3, which is all over the DNA stuff. Okay? After that, then it's all your projects. Okay? I'll start off the project sequence by going over the topics that we haven't yet covered. And I'll try and summarize those in as short a attempt as I would expect you guys to do for your projects, okay, for your presentations. So we're looking at 5 weeks before projects actually need to start happening, okay, just to give you a heads up.
And the paper is it next Thursday?
Whatever the syllabus says is when it's supposed to be due. Yeah. I can look up the syllabus and then I can tell you, but it's in the syllabus. I think it's next week.
Yeah, it's next week.
Yeah.
So it's due next Thursday, but our presentations anything after that?
Are going to be after that, yeah. So the intent is that you finish the paper so that you can then work on putting together a presentation that's reasonable. Yeah, because if the presentations and the paper are due at the same time, one is going to suffer. So the hope is you can finish the paper and then organize how you would present that. Okay, find all the pictures that you would end up using. Okay? Yeah,
Use
pictures.
on our essay.
That's fine. You can use pictures in your essay. I need words too, though. But yeah,
You figured one.
big pictures. No. So
Page 3, picture.
our nucleotides, or our DNA, we have varying stages of how these get processed. So we've got our nucleobases, our nucleosides, and then our nucleotides. The nucleobases can be categorized into two separate sections. You've got your pyrimidines and your purines. The pyrimidines have one ring, and your purines have two rings. Okay. In theory, you have seen this all before, yes. Okay. I would assume you've had some discussion on DNA. What we're looking at is then the subcategories of these. You've got your pyrimidines as cytosine, thymine, and uracil. What's the big difference within those? It's also up there. We can really only use one set in DNA and one set in RNA. So your cytosine is really only used in, is used in both DNA and RNA, which is listed at the bottom. Your thymine is typically only shown in DNA, though it can pop up in your RNA. And then your uracil is only used in RNA. Okay? So realizing when you can use these is going to become, in theory, somewhat important. When we look at the purines, we get slightly larger structures with those two rings, and we again get two different types. You get adenine and guanine. What's nice about the purines?
They're used in both.
They're used in both. So really the only characteristic difference that you need to remember is dealing with your thymine and your uracil. If you remember your thymine is DNA and your uracil is RNA, you should be pretty much set. Okay?
Uracil just takes the place of adenine?
Uracil takes, if it takes the place of adenine, then we've got the same base in there. What does uracil take the place of? Thymine. Okay. So uracil and thymine are ultimately the ones that are interchangeable. Why might that be the case? Yeah, take a look at their structures. Their structures are almost identical. Okay? The only thing that's really different between your thymine and your uracil is that your thymine has an extra methyl group on it. Okay? That methyl group is different enough to cause it to only be involved in one of those systems versus the other. We might see some overlap because that's really the only difference. We aren't going to see uracil showing up in DNA because that methyl group does have a significant change in how it interacts with water and other surfaces. Okay? If we go through and take a look at each of the other, or for instance your cytosine, you'll notice that we do see a significant difference in the structure. Instead of it being carbonyl coming off of carbon 4, we have the amine. That's going to have a substantial difference in its ability to interact with another molecule. Okay? In the case of the carbonyl, it's just an electron donor, okay, or a hydrogen bond acceptor. In the case of cytosine, it's both an electron donor, which makes it a hydrogen bond acceptor, and a hydrogen bond donor because of the hydrogens attached to that nitrogen. Okay? So we do see a significant difference in the overall properties between cytosine and the other set, our thymine and uracil. Yeah?
I need to go over like how GCs have 3 hydrogen bonds compared to
Yeah.
2.
Before we can get into the actual interactions, we've still got to talk about their overall structures. Okay? So those are referred to as your nucleobases. Okay? So that is, if we say adenine, that's the exact structure we're referring to. If we're going to refer to what actually gets incorporated into DNA, it's not just adenine. We have to include other things in it. What other things might we include? What does DNA stand for? Deoxyribonucleic acid. What we're referring to, this is our nucleo portion, okay, for our nucleobases. The ribo is going to be the sugar. Okay, so we at least need a sugar to also be incorporated when we're putting together our DNA. So just the nucleobases, yes, they can hydrogen bond with each other, but if those structures are freely tumbling, there's no way to guarantee that those hydrogen bonds stay permanently attached. Okay, with its proper matchup, not to mention its ability to actually propagate downwards or upwards, okay, continuing the chain. All right, so we need some way to actually lock those nucleobases in a position that allows for that hydrogen bonding or that specific hydrogen bonding interaction. Without that locked position, we're not going to get a standard storage for data. So what happens when we move up to the nucleosides? Okay, when we move to the nucleoside, we're going to take a base and we're going to fuse it with a ketopentose. Okay, why do I say ketopentose and not say deoxyribose? We're looking at our nucleobases and in particular our nucleosides. What we're concerned about is where these things are used. Where are they used? DNA and RNA. Do they use the same sugar? No. However, the sugar that they use is a ketone, and it does have 5 carbons. So we can refer to it as the ketopentose. We can actually use the one case where we're actually consistent with our nomenclature. Okay. The 2 ketopentoses that are most commonly used, as far as we're concerned, is our deoxyribose. Beta in reference to, what does the beta mean in our deoxyribose? That's our deoxyribose sugar. Our OH, it's not just that the OH is up.
Oh, it's on the same side.
It's the OH is on the same side as our CH2OH. Okay. Our last stereo center. Okay, what's the D in reference to?
What's located on the back end? The oxygen on the right?
The oxygen on the right is our anomeric carbon. That's the beta. What's the D in reference to? It's our leftmost carbon. Our penultimate carbon has the OH group on the right-hand side in the Fisher projection. Okay, so that's what our D was in reference to. So it's in reference to that carbon. Okay. If our CH2OH is aimed upwards, then we're going to have our D sugar. Okay. What's the 2 in reference to? It's referencing our second carbon. Where's our second carbon? Where's our first carbon? Let's try that.
It's a major substituent.
The first carbon
The one right after the.
is the anomeric carbon.
Right after the oxygen.
Yeah, I heard a couple answers, like one right after the oxygen. There was another one in there that was similarly correct, but just not quite enough information. Our first carbon is our anomeric carbon. Where's our second carbon?
The non-anomeric.
We're still.
I deserved it. I deserved it. That was a fair answer. 2, 3, 4, 5. If we take a look at the second carbon, what's special about that second carbon? Particularly when we consider sugars, where our sugars are supposed to have an oxygen on every carbon. It doesn't have an oxygen. It is deoxy. We've removed the oxygen. So the 2 is in reference to the fact that we no longer have the oxygen at that 2 position. That's where we're getting the deoxy from. Okay. The rest of the sugar is then a ribose general structure, so we can leave that as a ribose name. Okay. What happens if we move to RNA? We still have the oxygen on there, so we don't have to worry about the deoxy. It's still going to be beta, it's still going to be D, and we're still looking at a ribose sugar. So when we move to RNA, that's what we have on our leftmost side. We're looking at our beta D ribose. So this is our RNA sugar. And in the other case, we're looking at our DNA sugar. Okay, so slight tweaks to the overall structure. And if we look at the name for RNA, its full name is ribonucleic acid because the ribose, we have a ribose sugar in the RNA structure. Okay. The linkage between your nucleobase and the, what are you guys doing? The nucleobase and the sugar, just picking a lint string off here. The linkage between the sugar and your nucleobase is going to be at your first carbon on the sugar. So we're looking at attaching to the anomeric carbon, and it's going to go to either the first nitrogen in your pyrimidine or nitrogen number 9 if you're looking at your purines. Remember, your pyrimidine was the single ring, okay? And when you go through to number that or name that according to organic chemistry rules, that first nitrogen is what gets labeled as number 1, or the one that's labeled as 1. So in our pyrimidines, we get that nitrogen linking up. In the case of the purines, again, if we had to go through and number it according to our organic chemistry rules, we'd be linking up with nitrogen number 9, okay, or atom 9 within the ring, okay, which is that nitrogen. That's how we end up putting those two together. What type of linkage is that? It's not really an amide. Glycosidic is where I would go. We're looking at the linkage between the sugar molecule and something else. So it's some kind of glycosidic linkage. Okay? Man, the cough just won't go away.
Need some Teflon pearls.
Not even going to respond. Questions about our nucleosides? Sorry.
It's medicine.
Oh. So what we can then do is move to the nucleotides, which is ultimately what gets incorporated into DNA. The difference now is that we will now add a phosphate to the nucleoside. That phosphate is what's going to allow for our linkage through the DNA backbone. Okay? So our nucleotides are built of our nucleobase, followed by the sugar, followed by the phosphate group. There's two common places that we can put that phosphate. Okay, what's the structure of a phosphate? P with our oxygens linked around it. What's the reactivity of our phosphate? Very high. How? What's the reactive site of the phosphate? Okay, the oxygens are indeed negative. That's actually not going to be the source of reactivity because those oxygens can actually push their electron density into the structure. It's the phosphorus. What's special about the phosphorus? It's very partially positive, which means the phosphorus acts as we have an electrophile. What acts as our nucleophile? We're going to need another oxygen to share electrons in with it, okay, to form that bond. And what we'll end up eliminating in the process of that is one of those oxygens with two hydrogens, and we end up eliminating H2O. We end up losing water in the formation of that bond. So we could go back to our sugars to decide, okay, where do we have oxygens? Okay, I would start with the deoxy form because why is the deoxy form easier to work with than our, where'd it go? Oh, never mind, these are all deoxys. Why is the deoxy easier to find the reactive oxygen than the ribo form? How many oxygens are in the deoxy?
One less.
Yeah, there's one less. There's one less to worry about. So if we look at the deoxy form, we really only have two reactive oxygens where we can put the phosphate group. Those reactive oxygens are either going to be located at this position, which is carbon number
3.
We're looking at carbon number 3, Or if we push out, we've got the oxygen coming off of our CH2OH, our last carbon, which is carbon number 5. So when we're looking at our deoxy sugars, there's really only 2 positions that we could possibly put the phosphate, carbon number 3 and carbon number 5.
We should make a mambo number 5.
Feel free to do that. You can make that as part of your presentation. Okay. So what we're linking is putting a phosphate on at each of those positions. Why is that going to become particularly important that we have 2 possible places we could put the phosphate? What happens in DNA? This is a deoxyribonucleic acid. Why? Or is this the end of our DNA? No, we link a whole bunch of other of these on here. So this is just the monomer of the polymer of DNA. To form a polymer, how many bonds do we need to have come off of our polymer? It needs at least 2, otherwise we can't link to other monomer units. How many bonding sites do we have in DNA?
2.
We have 2, so we can actually link out. We need that at least 2 positions to allow for the elongation of our chain, or our polymer chain. Okay. When we move to RNA, what makes RNA a little bit trickier? We have another oxygen at carbon number 2. Okay. So why do we not look at putting the phosphate at carbon number 2, even when we move to RNA?
It's not as stable.
Why is it not as stable?
Because it's kind of actual. I don't know, we use like the.
I may have put that oxygen in the wrong spot too.
No,
Anybody remember? Did I put it right?
you did.
Oh, good. Why do we not put the phosphate on carbon number 2? We're just guessing actually at this point.
Well, because you're linking that sugar. Either the
I could
sugar-
just as easily link 2 to 5 as I could 3 to 5.
So it's the way that they build.
So it's going to be somehow to do with the structure. So we could look at a 2,5 and maybe build that structure, and maybe what happens is that kink, forcing it to twist, is not going to allow for the proper alignment of your nucleotides. So it may not allow for a nice sync up. One other option we could run with, which is not a good option in my opinion, if DNA runs 3,5 and RNA is supposed to mirror it, it should probably also
B35.
3,5. Okay? Otherwise, you're not going to get a nice mirroring image. Why is that argument not very good?
There's something viruses, it's just RNA.
That kind of works. That's where we were going with that. Did we start with DNA? If we think back to the origins of life, DNA is not considered the precursor to life. You're looking at RNA or proteins. Okay, I think the common thought now is RNA is the one that actually started life. Okay, so RNA should not be mirroring DNA. It should actually be the other way around. DNA should be mirroring RNA. So it is very likely that what we see on that 3,5 linkage is because if we try to create a polymer chain from the 2,5 linkage, we're probably not aligning those nucleotides in a lowest energy state. Okay? Typically, when we're looking at our nucleotides, they form in the 5 position. Okay, why might we link them in the 5 and not the 3 first? Again, just kind of guessing.
It's more reactive
Why is it more reactive?
because it's so positive.
I don't see the positive. We're referring to the oxygen. The oxygen is actually going to be negative in this case. Why is the 5' oxygen more reactive than our 3'?
It's way off.
Why is it way out there?
Because it's attached to
Off the ring.
a branch of
Yeah. Our 5' is actually attached off of the ring. Our 3' is attached to the ring. Sterically, the 5' is probably more accessible than the 3'. We're probably looking at a steric argument to suggest why the 5' is actually the preferred phosphate state on its own. Okay? So that's probably one of our reasons there. Okay, so our 3', while we can put a phosphate there, is really only available for the polymerization, our chain lengthening, adding multiple nucleotides in there. The 5' is where we're going to start at our primary state. Okay? One extra statement here. You notice that I've got 5 with a dash, or a, what is that, apostrophe? 3 with apostrophe, and I keep calling it 5' and 3'. What's that prime? What's that apostrophe representing? This actually goes into nomenclature, organic nomenclature. It's really advanced organic nomenclature, so I'm not surprised that you don't know it. We're looking at linking multiple rings with each other. Okay, we've got our purine ring structure linked to another sugar ring structure. If we've got ring structures that are not fused, so in the case of our purine, we have 2 rings that are fused to each other, so that counts as one ring structure with our purine. Okay? So if we connect them through a sigma bond, we now have 2 rings. Okay? Which means, how are we going to go through and name it? We're going to decide that one of those rings is our primary ring. One of those rings will be our secondary ring. The primary ring we number as normal, 1, 2, 3, 4, etc. Our secondary ring will still number as normal with our 1, 2, 3, 4, but we have to come up with some way to say this is our secondary ring. We do that with the prime. Okay? So that's what that prime is floating out there for. It's just referencing the fact that you're referring to a secondary ring, if you will.
Why wouldn't that make no sense?
Why wouldn't that make no sense?
No, why, and then comma, that doesn't make any sense.
Which
If
part?
it's prime, why not primary? That's the first thing you-
That might be the first thing that comes into your head, but what happens if we have just one ring? Do we refer to that as a primary ring, or do we just number it as normal? We number it as normal. So it's really almost like a zeroary ring and our primary ring that comes after it. So the prime is only included when we're referring to at least a second ring. Okay? Yeah, you're right. The reference as prime is a weird response, but all we're really saying is it's an extra one. Is that a hand, or you just stretch?
Three rings.
What would end up happening with 3 rings?
2 apostrophes?
Double prime.
What is it?
How many rings do you want to attach? In theory, if you're going to go through and name it, you're going to have to name each of those as a new prime ring. Okay? Other questions on that? Okay, so now what we've got is our nucleotides. Now what we're concerned about is now putting these together and actually making our DNA structure, so building this out. So a couple things that we can look at for our shorthand. You'll notice that we've got polymers in the bottom half of this. Okay, so those are going to be our polymer sections where we've actually linked multiple nucleotides next to each other. What's written above is another notation. What is that notation referencing? What does the A stand for? Is it really adenine? Adenine is just the nucleobase. So we're actually scaling it up further. We're moving it up to the nucleotide, and it's not officially adenosine either, because adenosine is just referring to the which is the next one up? I always forget this. So we got nucleoside. Sorry. Adenosine is now just referring to the nucleoside. How do we get to the nucleotide? What's the difference between a nucleoside and a nucleotide? The phosphate. There's our phosphate. So what we're looking at is our nucleotide. How would we name that nucleotide? Phosphoadenosine, because it has that extra phosphate. Where is that phosphate located? At position 5 or position 3 on the adenine? I'm pretty sure I gave you the answer there. Okay. We have to come up with a standard notation so that when we put that phosphate on there for our P, that we have to know which linkage it's going to. Because our 5' is the primary place to put the phosphate, we'll label that phosphate on the left-hand side coming first being our 5'. If you see it marked to the right, what we're saying is that phosphate is located on the 3' end, not the 5' end. Okay? So in the case of this first one, we have a phosphoadenosine with the phosphate being on our 5' side. So we could say 5' phosphoadenosine. What about the next one? It's going to be a 3' phospho.
Thiamine.
Good question. You can't say tyrosine.
No, it's
Actually, do we just say thiamine?
yeah.
That's just the base, though. Huh. Well, there we go. Phosphate, anyways, is on our 3' side. Let's focus on that. And the next one, where's our phosphate?
I thought the phosphate.
So it can always be on the 3 or 5'. It is most commonly found on the 5' side. Okay? Just because it's most commonly found there doesn't mean that when we come up with a notation that it's always there. Okay? What happens when we move to now polymers? Well, to link up our polymers, it's the phosphate connecting to the 5' and 3' of two different positions. So we would start with a phosphate on the 5' side of our guanine, right? Close enough. Followed by a phosphate on our 3' side, which then follows into the 5' of our adenosine, which then has a phosphate on the 3' side, which then follows into the cytosine.
But if you were to split it, it wouldn't.
So if we were going to go through and break it, so that's, again, beside the point. If we're going to break, we're still going to go through and break where we have the most common. We would expect the phosphate to stay with the 5' side. But we have to come up with a nomenclature system that allows us to say that it could be in the other place, just in case. So that's all it ultimately is. What you would expect to see is the phosphate always on the 5' side, on the left-hand side of your nucleotide. Okay? When we go through and make the polymer, we would then link it up. Okay? You'll notice that the one big difference with this polymer is we have a phosphate on the left-hand side, and we don't have a phosphate on the right-hand side of our polymer. Why not? Because you typically don't put a phosphate on the 3' side. Okay? The phosphate that linked the A to the U came from our uracil, okay, on the 5' side, linking up to the 3' of our adenosine. Kind of makes sense? You'll notice that brings in a lot of P's, which is kind of irritating. So we can come up with a different format. What we can do is assume that it's always going to be on our 5' side, and that we will start our DNA chain listing 5' to 3', which means when we list the next sequence, our GACT, the G is located at which position? Our 5', which means there is a phosphate implied hanging off of that. And our thiamine is at the 3' end, and we would not expect to find a phosphate out there. We would expect that to be blank. Okay. Where do we find GACT?
DNA.
How do you know it's DNA?
Is that what
There's
you?
thiamine on here.
It's got our thiamine and uracil. Remember, thiamine could show up in theory in both, but if we're going to CT, more than likely we're looking at DNA. What about our first sequence?
RNA.
Expect to see that in RNA because we've got the uracil. What about the last sequence? Could be either. Okay? Because our adenine, cytosine, and guanine show up in both sequences. So without any other information, we don't know if this is DNA or RNA. Okay? How could we change this structure to help us clarify that it's DNA without adding thiamine? What's the difference between DNA and RNA?
Single-stranded or double-stranded?
We haven't even talked about double-stranded yet, so we can't get away with that. The sugar residue. What's the difference between our sugar? One is a deoxyribose. What sequence do we have now? We have DNA because we have the D representing the fact that we have a deoxy carbon chain. So you might also see a D listed in there as well. Okay, just want you to be aware of all the possible shorthands that can go with this. More than likely, the shorthands that you will end up seeing and working with the most commonly, I guess it doesn't really make sense to box that in. Okay, would be that upper right-hand sequence, or even our lower left-hand without the D. Okay? Questions about the shorthand and our general primary sequencing of our DNA? Okay. What we can then do, we're not finished with primary structure. With our primary structure, we can then propagate this outwards, and we can link our phosphates to each other through the 5' and 3'. So if I circle this phosphate, you all see that phosphate? Make it red. That phosphate. What amino, or sorry, not amino acid. What nucleoside brought that in? How do you know it's cytosine?
Because that's the one that is attached to the 5' end.
We look at where the phosphate's attached. Cytosine is where the phosphate's on our 5' end. The cytosine is what brought in that phosphate. Okay. The linkage between those is referred to as the phosphodiester bond. Why would we call it a phosphodiester? We have a phosphate group that's giving us our ester functionality, so the carbonyl. It's a diester because we have an oxygen on either end linking up our carbons. Okay, so we have that phosphodiester, which is ultimately what's making up our backbone as well. So we have the phosphodiester backbone. When we're looking at DNA, remember we're dealing with the deoxy ribose. You'll notice again that we should usually drop in that 2' to make reference to the fact that we're referring to the secondary ring within our sequence. Oops. Any questions on that? We just talked about all of that. DNA, secondary structure. So when we move up to the secondary structure, it turns out DNA comes in as a double helix, which in theory you all probably have heard of. And what we're looking at is an ordered arrangement of these nucleic acid strands. So the Na there is in reference to our nucleic acids. The double helix was kind of developed and put together by a variety of people that contributed to this. I got actually really angry at the textbook for a while and stopped working over spring break because I didn't see any reference to Watson and Crick, which I thought was really weird. They did actually reference Watson and Crick. I just can't read right. So Watson and Crick are the ones that were given the Nobel Prize for identifying that DNA is a double helix. Okay? So they're the ones primarily responsible for that. Okay? How did they come up with that information? Depends on who you want to talk to, on exactly how that whole debate came about, which is also why I was surprised that Watson and Crick only showed up once, and Rosalind Franklin's name didn't show up at all in the textbook, which I thought was even more interesting. What did Rosalind Franklin do? She set up your X-ray crystallograph. Okay. And it was that crystallography or that graph that Watson and Crick, in theory, were able to interpret to come up with the double helix analogy. There's another name, which for some reason doesn't look right to me, but maybe I'm right. I think that's Chablis, maybe? I think that's how it's spelled. I'm afraid that I used the wrong name. Was also a big contributor to the overall analysis that Watson and Crick had to come up with. His big contribution had to do with we had equal amounts of A and T and C and G. Okay. So he was actually able to design experiments that proved, regardless of the source of the DNA, that you had roughly equal amounts of adenine as you did thymine, and you had roughly equal amounts of cytosine as you did guanine. Why would that be important? Those are complementary base pairs in the double helix. If we didn't have that double helix, we might not expect to see that complementary base pairing. Okay? So that was an extra piece of evidence that Watson and Crick had at their disposal to go through and kind of compile and put this all together. Okay? So you can come up with any argument you want on deciding whether or not they stole people's information. Ultimately, what they were able to do was take the information that was available and put it all together into one happy puzzle. Okay, they just happen to be the first person or first group of people to do that. Okay, without necessarily having to do a bunch of extra research on their own. So really you can blame everybody else for not getting that last analysis step. Okay. Yeah, you can blame everybody else for sharing and Watson and Crick for not sharing. So yeah, there's all sorts of debate on that. I'm sure you can read up on that more if you'd like. Okay, but those are kind of your primary contributors to the conclusion that we have a double helix. The strands will align in an anti-parallel fashion. So if we look really carefully at both the top and the bottom, you'll notice that the 5' end of our green strand aligns up with our 3' end of the yellow strand. Okay, so you can check both the top and the bottom to see that alignment. So what we've got is our DNA strands running in opposite directions, almost like a beta or an anti-parallel beta sheet. So we're seeing this anti-parallel alignment of our DNA strands. The other stabilizing factor is that each of those amino acid pairs has hydrogen bonding. That hydrogen bonding effect is really what's helping to contribute to the DNA actually staying bound to each other. The helix is due to the fact that we have to then align those properly due to the steric arrangement of the backbone. Okay, so kind of neat. We've got that DNA secondary structure. Again, in probably a more advanced class, you'd be responsible for recognizing all the individual pieces coming off of this. What other things could we look at or quantify? The image gives you some of it.
There's like some DNA there,
We don't even want to get into that yet. Just looking at the structure itself, okay, we could look at the number of amino acid pairs per turn. Okay.
Minor versus major group?
We can look at the minor groove and the major groove. When you take a look at this, you'll notice that the distance between our green and yellow pairing, well, let's just make it bold, there versus our distance over here is substantially different. So we end up with a minor groove and a major groove, the minor groove being the smaller of the two, hence minor, and our major groove being the bigger one. Okay. Why might that become important?
There's enzymes.
Yeah, there's enzymes.
Enzymes can access the major groove easier. We may have enzymes targeting minor or major grooves depending on what they were designed to do. Okay? We could take that further because that's a natural pathway. We could also look at unnatural pathways if we're going to look at mutagenesis. If we were going to somehow cause the DNA to be destroyed, okay, or make some chemical change to it that prevents one of the enzymes from working, we would probably target something within the major groove because it's going to be more accessible and more available to actually drop something in there. Okay?
Make it a triple.
I don't even know what to say at triple helix.
Well, there's left-handed and right-handed too.
And we aren't even at that yet, but you're right. Within this standard form of DNA, we can look at the size of your large groove and your small groove. So that's our major and minor. They've quantified numbers on this figure. Your minor is looking at 12 angstroms, and your major is 22 angstroms. Okay, so that gap spacing is something that we could evaluate and have you memorize. I think there was something else that's important. In theory, I've got listed on the next page, or one of the next pages. When we're looking at our base pair alignment, so this was Chablis or Chablis, or however you want to say his name's kind of setup, is that we saw equal amounts of adenine as thiamine and equal amounts of cytosine as guanine. Okay, which would suggest that they have to be interacting with each other, and that's why we're getting that equal ratio. When we look at the double helix, what we end up seeing is some nice hydrogen bonding interactions between those pairings, and if we tried to mismatch those, we don't get as nice a pairing alignment, which is why we see A always bound with T, C always bound with G. Okay? When we're looking at our adenine and thiamine interaction, we have just 2 hydrogen bonding interactions. When we move to our cytosine and guanine, we're looking at 3 hydrogen bonding interactions. Okay? What do you think would happen to the melting point if you had a lot of C and G?
Higher melting point.
Why should I have a higher melting point?
Because it has 3 bonds.
It has 3 hydrogen bonds that we'd have to break. So if we have an increased amount of cytosine and guanine within our DNA double helix, we would expect it to require a lot more energy to break that apart because we have a lot more hydrogen bonding interactions. Okay? Everyone pretty much remembers or has some knowledge of that kind of hydrogen bonding base pairing within it. But there's a couple other interactions that we have to look at as well. You get something known as your base stacking, which you may have heard in organic chemistry as pi stacking. Okay? That pi stacking really plays an important role in organic with what reaction? Diels-Alder reactions. The Diels-Alder reaction, one of the reasons to explain why we get the particular alignments that we do within our products has to do with the pi orbitals overlapping with each other. If we take a look at our base pairs, we notice that we've got a lot of pi system within it. In fact, all of them have pi systems all the way through them. Those pi interactions, when we stack the DNA on top of each other, actually contribute to a stabilizing effect. Okay, so we can reduce the overall energy of that pi system by stacking it on top of other pi systems. Okay. What's an interesting effect of having these stacked pi systems?
Wow.
Sure. I don't know how to respond to that one. I don't know that answer. You've got a sea of electron density stacked on top of each other. What does a sea of electron density typically allow for? Passage of electric current. We could potentially use DNA as electric wiring, okay, because of that sea of electron current between it. Still active research, but we have the potential for something like that. Okay. One of the other kind of effects that you're going to see in this is that we get a slight propeller twist to the stacks. So we don't have a perfectly flat alignment of those hydrogen bonds. They actually twist a little bit off of each other. Why would they twist? When we look at the minor groove, water can potentially get in there and interact with our amino acids. I keep calling them amino acids, nucleic acids. Most of those nucleic acids are pretty hydrophobic. Really, we only have that hydrogen bonding interaction within the middle. The rest of it is pretty hydrophobic. Okay? So if water makes it into the DNA structure, what's that going to do to the double helix? Destabilize it and prevent it from actually forming that nice stable structure. So that could be a bit problematic. So what ends up happening is it does this little propeller twist, okay, which does weaken some of the hydrogen bonding interactions, but at the same time it strengthens the hydrophobic interactions of the pi stacking system. That strengthening of your hydrophobic system now actually allows for a more stable DNA structure and less likely for water to intercalate into the DNA base pairs. Okay? So DNA isn't quite as simple as just stacking those amino acids through it. And this is what Victor was starting to get to. We get a variety of helices. They aren't all the exact same. We get 3 primary types. You get your B DNA, A DNA, and Z DNA. Your B DNA is considered the physiological state, the most common one. And so when we talk about DNA, that's usually the one that we're referencing. Okay? So that's where we have the right-handed helix. The previous slide, we were looking at roughly a 20 angstrom diameter. That's referring to the entire diameter of the structure. The inner diameter, so where all the nucleobases are located, is about 11 angstroms. What's the distance from one nucleobase to the other? 11 angstroms. So that helical shape in bDNA perfectly aligns the amino or the nucleic acids to actually match up and set up a favorable hydrogen bonding interaction. Okay? So the structure of bDNA actually helps with our hydrogen bonding. We're looking at roughly 10 base pairs per turn, and this is our 34 angstroms for a repeat within the helix. Okay? aDNA has not been found in nature. What they believe it to be is an artifact of DNA We're pulling it out of its natural state. We're removing potentially a lot of the things that stabilize and contribute to its folding structure, and that destabilization leads to a slightly different form, and that's our aDNA. Okay? So we're still looking at a right-handed helix, but it's a little bit thicker than bDNA. We're looking at 11 base pairs per turn. Okay? So we're kind of elongating it a little bit so that we can squishing it so we can put some more base pairs within each of those rotations. Okay? The last form is zDNA. Still don't have exact ideas on what's going on with that. It's primarily considered to be for gene expression. We're looking at a left-handed double helix. What's in reference to left-handed versus right-handed? Has to do with the rotation. If we use your right hand, my thumb is pointing upwards, right-handed DNA coils towards the right. We use left-handed DNA and we go on looking at that pointing upward direction, it coils to the left. Okay, so that's where we're getting our reference of left versus right-handed DNA. Think about a screw. Okay, it's threaded only one direction. And if you try to screw it left, it's never going to go in, okay, unless it's reverse threaded. Right? Your DNA has a very similar idea. So if we're looking at right-handed DNA, our hand would follow around the structure upwards. If we looked at left-handed DNA, we wouldn't actually be able to put our hand on it with our thumb pointing upwards to actually get it to fit in the major groove. We'd have to turn the hand over. Okay. Your zDNA is your left-handed, and then it also usually consists of alternating base pairs. Okay. So instead of having groups of, say, all C, or all G, or all A, or all T, what we'll get is an alternating system. Okay. It just happens to be one of the things that they noticed when they looked at zDNA. We've got some pictures of those DNA structures on the right. There's no way I'm going to put that on a test, but I might go through and ask on kind of an open-ended question. There are 3 forms of DNA, or 3 known forms. Compare and contrast. Okay. So that would be where you would effectively list everything on this slide. Okay. So that is our secondary structure of our DNA. Okay, so we just get with these double helices. We can then push this further, just like we did with proteins, and we can end up with tertiary structure. Within the tertiary structure, what's going to end up happening is we can actually crank the twisting down tighter on our DNA structure. So we end up with either a tighter coil or a looser coil, because we could potentially unrotate that helice. Right? In the process of doing that, we can either end up with a positive supercoil or a negative supercoil. Okay? If we go through and do that rotation, particularly in the positive supercoil case, in the case with histones, what ends up happening? You end up generating these extra loops within it as we put in more and more rotations. Those loops then get stabilized by, I've been dying to say it, Braden. We're looking at histones. Why do you keep changing? We're now getting histones. Our histones are going to come in and help stabilize and maintain that supercoiling process. Okay? We get it both, this supercoiling process happening both in prokaryotes and eukaryotes. The difference between our prokaryotes and eukaryotes is either circular versus single, or not single-stranded, straight chain. There we go, straight chain. In our prokaryotes, we get the circular DNA, so it does loop back on itself. In eukaryotes, we have the straight chain.
Does the pi overlap have to do with how tightly or untightly it's wound?
The tighter you wind it, the more you're going to get a pi overlap, for sure, because you're bringing those amino acids, those nucleic acids closer to each other. Okay, so you would increase the pi stacking within that.
Does that stabilize it when it gets tighter or no?
Yes.
Yes and no. So it changes the overall properties. Okay, so we can either supercoil that and then kind of what happens to your major and minor grooves in the process of supercoiling it. Let's look at it that way. What happens to the major and minor grooves when you supercoil? We're winding it tighter. It decreases that gap. Well, if the gap gets smaller, can things get into the DNA? No. So what's that going to do to its stability? Should increase the stability of the overall structure. Okay. If we unwind it, what happens in the unwinding process? Okay. If we go to a negative coil, okay, we're now going to actually unwind the helice and expose the nucleic acids, actually open up those grooves even more. In the process of opening them up, we're now going to expose them to potentially enzymes or other kind of reactive systems. Okay? This process of supercoiling is controlled by your topoisomerases. We're calling it an isomerase because we're really just changing the isomer structure of this. Okay? And so what they will end up doing is either coiling it tighter or looser, depending on what they were actually asked to do. These get separated into two classes, and I hate to admit this, but I don't quite understand the difference between those two classes because they don't seem to explain class 1 very well. In class 2, that makes sense. The class 2 topoisomerase completely cleaves the DNA and folds the other strand of DNA through that open gap. Okay? That's going to then allow for a tighter coil because we actually end up looping it tighter and tighter. Okay? Or we could look at the reverse, pull the DNA back through that open hole. The class 1, it doesn't completely cleave. It only cleaves one strand. So maybe what it does is goes one strand, pushes through, closes, and opens the other one.
Yes.
Yeah. So it still has to go through and do that cleavage. You just end up doing it in 2 steps. DNA gyrase is one of an example of those. Okay, it will allow that process to occur. Wow, we went from, it's just like yesterday, 5 minutes left to 5 minutes past.